Somewhere in your body right now, there are cells that refuse to die. They're not healthy. They're not doing their job. But they won't disappear either. Scientists call them senescent cells. Almost everyone else calls them something more memorable, zombie cells. And the more of them you carry, the faster parts of your body seem to age. What makes a cell become a zombie? To understand this, you need to know what a normal cell is supposed to do. A healthy cell has three basic options: keep dividing and doing its job. Or, if it gets damaged beyond repair, kill itself through a clean, orderly process called apoptosis. That second option sounds grim, but it's actually protective. A damaged cell that quietly disappears can't cause much trouble. Zombie cells choose neither option. They stop dividing, but they refuse to die. They just sit there, present but useless, stuck in a kind of biological limbo. This isn't rare or exotic. It happens to real cells, in real tissue, for reasons that are surprisingly well understood. How a cell ends up stuck. There are a few well-documented paths into this state. The first is telomere shortening. Every time a cell divides, the protective caps on the ends of its chromosomes, called telomeres, get a little shorter. Think of them like the plastic tips on shoelaces. Eventually, after enough divisions, those tips wear down too far. The cell senses this as a threat to its own DNA. Its response is to stop dividing completely, not to repair the problem, just to freeze in place. Here's a simpler way to picture it. Imagine a pencil that gets shorter every time you sharpen it. Eventually, there's nothing left to sharpen. The pencil still exists. You just can't write with it anymore. Your cells are a little like that pencil, except instead of quietly retiring, this particular pencil sticks around on your desk, in the way, doing nothing useful. The second path is direct DNA damage. Radiation, toxins, and ordinary oxidative stress can all injure a cell's DNA. When the damage is too severe to fix, but not severe enough to trigger cell death, the cell can lock itself into this same frozen state instead. Think of DNA as an instruction manual the cell reads every single day to know what to do. A few torn pages, and the cell can usually work around it, but when too many pages get damaged at once, the cell decides it's safer to stop reading the manual altogether. It doesn't throw the manual away. It just stops using it, forever. The third path involves the cell's internal stress signals. Malfunctioning mitochondria, the tiny structures that produce a cell's energy, can push a cell toward senescence on their own. So can certain early cancer-like changes inside the cell, which the cell may be trying to contain by shutting itself down before things get worse. In simple terms, mitochondria are like a cell's power plant. When that power plant starts breaking down, the whole cell runs low on energy and starts sending out distress signals. Sometimes those distress signals are enough on their own to push a struggling cell into this same stuck, non-functioning state. In every case, the underlying decision is similar. The cell detects a threat. Instead of fixing it or ending itself cleanly, it simply stops functioning and stays put. The part that makes them dangerous. Here's where the zombie comparison actually earns its name. A frozen, non-functioning cell, sitting quietly, wouldn't be a huge problem on its own. The real damage comes from what these cells do next. Senescent cells don't stay silent. They actively release a mix of inflammatory molecules into the surrounding tissue. Researchers call this the senescence-associated secretory phenotype, usually shortened to SASP. It includes signaling proteins that promote inflammation, along with enzymes that can break down the structural matrix holding nearby tissue together. Here's the plain version. Imagine one grumpy neighbor on an otherwise peaceful street who spends all day loudly complaining out their window. Their neighbors don't get physically sick from it, but the constant noise puts everyone on edge, wears down morale, and slowly makes the whole street a slightly worse place to live. That's roughly what a zombie cell is doing to the tissue around it. It's not attacking directly. It's just constantly broadcasting low-level chemical noise that wears everything down over time. This matters for two reasons. First, Chronic low-grade inflammation is already linked to a long list of age-related diseases, from heart disease to some cancers. Second, and more unsettling, this inflammatory signal doesn't just sit in one place. Research has found that these signals can push nearby healthy cells toward the same frozen senescent state. That's the part that earns the zombie name. It's not really about individual cells refusing to die. It's about one non-functioning cell quietly recruiting its neighbors into the same condition, spreading dysfunction outward like a slow, invisible infection. Why they build up as you age. If your body creates these damaged cells constantly, why don't they build up starting from childhood? The answer is that, for most of your life, they don't get the chance to. Your immune system has cells specifically responsible for identifying and destroying senescent cells before they spread trouble. Natural killer cells, the same NK cells that show up in a lot of aging research, are one of the main tools your body uses for this job. In a young, healthy immune system, senescent cells get flagged and cleared out fairly efficiently, often before they accumulate enough to cause noticeable damage. Think of your immune system as a cleaning crew that walks through your body every day, removing broken furniture before it piles up. When you're young, that crew is fast and thorough. Nothing sits around for long. The problem is that immune surveillance itself slows down with age, a process researchers call immunosenescence. As NK cell activity and other immune functions decline, the body's ability to clear out senescent cells declines right along with it. Fewer zombie cells get removed, more of them stick around. And the ones that stick around keep producing that same steady drip of inflammatory signal. Going back to the cleaning crew metaphor, imagine that same crew slowly getting smaller and more tired every year. Broken furniture starts piling up faster than it gets hauled away. Eventually, there's simply more clutter around than the crew can keep up with. This creates a feedback loop that researchers now consider one of the core mechanisms of biological aging itself. Senescent cells accumulate. Their inflammatory output damages nearby tissue and immune function. That damage makes it even harder for the body to clear out the next batch of senescent cells. Round and round it goes, quietly, for decades, long before anyone notices anything unusual. Studies looking at tissue samples confirm this pattern directly. Senescent cell burden is measurably low in young tissue and rises steadily with chronological age across multiple organs. This isn't a hypothetical trend. It's something researchers can count under a microscope. Where this shows up in real disease, zombie cells aren't just a background curiosity in aging research. They keep showing up in the specific diseases most associated with getting older. In blood vessels, senescent cells have been connected to atherosclerosis, the hardening and narrowing of arteries that drives heart attacks and strokes. In simple terms, imagine a garden hose slowly turning stiff and clogged from the inside. Blood has a harder time flowing through, and the heart has to work harder to push it along. In bones, they've been linked to osteoporosis, where bone tissue breaks down faster than the body can rebuild it. Bones are constantly being quietly repaired and rebuilt in the background. Zombie cells seem to slow down the rebuilding side of that process, so bones gradually lose density faster than they should. In the lungs, senescent cells show up in pulmonary fibrosis, a scarring disease that slowly reduces the lung's ability to move oxygen into the blood. Picture a soft, stretchy balloon slowly turning into something closer to stiff cardboard. It doesn't expand and contract as easily anymore. Perhaps the most striking research connects them to the brain. In studies of Alzheimer's disease, senescent cells accumulate around the same protein tangles and plaques long associated with the disease. Some researchers now argue that senescent cells aren't just a side effect of that damage. They may be actively contributing to it by keeping inflammation switched on in exactly the tissue that can least afford it. None of this means senescent cells are the single cause of these diseases. Aging is complicated, and most age-related conditions have several contributing factors working together. But the pattern is consistent enough that senescent cells are now treated as what researchers call one of the recognized hallmarks of aging, sitting alongside things like DNA damage and mitochondrial decline as a core driver, not a minor side note. the science that's trying to fix this. Here's where the story shifts from problem to potential solution. If senescent cells cause harm mainly by refusing to die and sticking around, an obvious question follows. What happens if you simply remove them? Researchers have tested this directly, using mice genetically engineered so that senescent cells can be selectively destroyed on command. The results have been consistent across many of these studies. Mice with fewer senescent cells show improved physical function, reduced frailty, and in several cases, a longer healthy lifespan compared to mice where senescent cells were left alone. That result sparked a new category of experimental drugs called senolytics. These are compounds designed to selectively push senescent cells toward the cell death they were avoiding in the first place, while leaving healthy cells largely untouched. Some of the most studied combinations include dasatinib paired with quercetin, along with a plant compound called fisetin, found naturally in strawberries and other fruit. In plain terms, think of senolytics as a very specific kind of eviction notice. Most cells never receive it because they're doing their job normally and paying their rent, so to speak. Only the stuck, non-functioning zombie cells are targeted for removal. In theory, that's what makes this approach appealing. It's not about killing cells broadly. It's about clearing out specifically the ones already causing trouble. In animal studies, these senolytic treatments have shown benefits across an unusually wide range of conditions. Improved bone density, reduced markers of frailty, better outcomes in models of lung scarring and muscle degeneration. In dystrophic muscle tissue specifically, clearing senescent immune cells has been shown to restore some function to muscle stem cells that were otherwise struggling. What's actually happening in humans? Now, the honest part. Almost everything described so far comes from cell cultures in a dish, or from mice in a lab. Human research is still catching up, and it's important to be precise about where things currently stand. Early human trials are underway, but they remain small and preliminary. One clinical trial protocol, run through a major research hospital, is specifically testing senolytic treatment in older postmenopausal women with elevated senescent cell markers in their blood, focused on outcomes related to bone health. Results from trials like this are still being gathered. Researchers working directly in this field have been candid about the challenges of moving from mice to humans. One recent review pointed out that early human studies reveal how varied the underlying aging process is from person to person. The same senolytic treatment might help one person meaningfully and do very little for another, depending on how much senescent cell burden they're actually carrying to begin with. This means future trials will likely need to identify which specific people have enough of a senescent cell problem to actually benefit from clearing it out. Put simply, giving everyone the same treatment is a bit like handing out umbrellas to an entire stadium when it's only raining on one section. The people sitting in the dry seats won't notice much difference either way. Researchers are still working out how to find the people actually sitting in the rain. Another honest limitation is more basic. Most existing senolytic compounds weren't originally designed for this purpose. Dasatinib, for instance, is a cancer drug being repurposed here. Long-term safety data in healthy aging adults, taken specifically for senescent cell clearance rather than cancer treatment, is still limited. Reviews in this field describe current clinical results as showing moderate efficiency so far, not the dramatic anti-aging breakthrough that headlines sometimes suggest. So here's the honest summary. The biology behind zombie cells is remarkably well mapped out. The animal data showing benefits from clearing them is extensive and consistent. But turning that into a proven, safe, widely available human treatment is still a work in progress, not a finished product. Why this matters beyond the lab. Even without a finished drug on pharmacy shelves, this research already changes how scientists think about aging itself. For decades, aging was often treated as an unavoidable, uniform process. Everything just slowly wears down together. Senescent cell research pushes back against that idea. It suggests that at least part of aging comes from specific, identifiable cells actively causing damage, rather than everything simply fading at the same steady rate. That distinction matters, because it reframes the goal. Instead of trying to slow down every part of aging at once, researchers can target one specific, well-understood mechanism, remove the cells causing the inflammatory damage, and see whether the tissues around them recover function they had already started losing. This is also where earlier ideas from this channel connect back together. Anything that supports a stronger immune system, especially stronger natural killer cell activity, may be doing double duty here, not just fighting off infections more effectively, potentially clearing out more of these zombie cells before they get the chance to spread dysfunction to their neighbors. What you can actually do right now. Given that senolytic drugs are still experimental, what does this mean for someone watching today, right now, who isn't enrolled in a clinical trial? First, understand that your immune system is your existing zombie cell clearance system. It's already doing this job, just less efficiently as you age. Anything genuinely shown to support immune function, from adequate sleep to regular movement to managing chronic stress, is supporting the same clearance process this entire video has been describing, even without a specialized drug involved. Second, with Chapter 2 of supplement marketing that borrows this science without earning it. Fisetin and quercetin are natural compounds, and you'll increasingly see them marketed directly to consumers using the word senolytic on the label. The laboratory doses used in published animal studies are often far higher and more concentrated than what a typical supplement capsule provides. A compound working in a controlled lab experiment is not automatically the same as a bottle on a store shelf delivering a meaningful dose. Third, keep an eye on this research rather than acting on it prematurely. This is a genuinely fast-moving field, with human trials actively underway right now. The responsible position today is informed patience, not early adoption of unproven interventions, and not dismissal of a mechanism that's backed by a large and consistent body of evidence. The bigger pattern. There's something fitting about ending here on a mechanism most people have never heard of, sitting quietly inside cells that refuse to either work or die. This channel keeps returning to a similar idea from different directions. Aging isn't one single unstoppable process rolling forward at a fixed pace. It's made up of specific, identifiable pieces: a dulled immune system, a brain losing its regular exercise, and now, a small population of stubborn cells refusing to leave the stage after their part is over. None of these pieces are simple to fix, but naming them precisely is still progress. You can't target a problem you can't describe. For a long time, aging itself felt too vague to target directly. Zombie cells are one small, specific reason that might be starting to change. Think about it this way. A doctor can't treat getting older as a single condition, but a doctor can, at least in theory, treat a measurable pileup of specific dysfunctional cells in a specific tissue. That's the quiet shift happening in this field right now. Aging is slowly being broken down into smaller, more specific, more workable pieces. Zombie cells are simply one of the clearest examples of that shift so far. If this changed how you think about what's actually happening inside your body as you age, subscribe. Until next time, take good care of yourself.